Many visitors to the Peabody Museum don't realize that what we have on display in our public galleries is less than 1% of the museum's collections. I'm Tim White. I'm one of the collection professionals here at the museum. Today, my colleagues and I are going to take you on a behind-the-scenes tour of the Peabody to see many of the specimens and artifacts that people rarely get to see. Some people think that the Peabody is all fossils and bones, and we certainly have our share of those. But we have also collected everything from arrowheads and pottery to plants and birds, fish and butterflies, and even DNA. And a lot of it is housed right here in a complex that includes the museum, the Class of 1954 Environmental Science Center, and the Klein Geology Laboratory. While we already moved most of our anthropology collections to West Campus in 2008, we still have some important artifacts waiting to be moved from the Peabody basement. Let's meet Maureen DeRoss, one of our anthropologists, to learn about some of these artifacts. Maureen, could you tell us a little bit about the contents of this room?
This is our Archaeology of the Americas room, and it's North America all the way through South America. And we have collections such as ceramics and lithics, and here's some of our earliest collection, and this is a tripod vessel from Panama. This was used as an offertory vessel, probably found in a funerary context. This was actually part of one of our earliest digitization projects as well, where 30,000 whole vessel or figurine items were digitized, photographed by graduate students whose specialty was South America.
How many objects are in this room?
This room has it's over 40,000 objects. It's closer to 70,000 objects. I mean, some of them are the larger whole vessels or stone carved objects, but a lot of them were excavated material that are a lot smaller, but all needs to be inventoried, digitized, and photographed before we can move them.
What's along the wall, Maureen, behind all of the glass?
Along the wall is mostly our whole vessels from Peru. One of our largest South American collections is from Peru. We've had several archaeologists over the years, including Wendell Bennett, who was interested in the Wari culture. But we also, right here, we have some moche vessels. And it's simply, we didn't want them in movable storage coming out of a drawer, so they're on shelving. It's a worldwide collection. We have a large collection of North American basketry, and we have the largest Caribbean archaeology collection in the world. And we have large collections from the Pacific. So these are large wooden poles and basketry fronts that decorated the South Pacific houses.
And where are we moving them to?
We're going to be moving them to our facility at West Campus, where we already have 75% of our collections, and we're hoping to be able to have more browsable, accessible storage.
I've been managing collections for over 30 years, and one thing I've learned about improving the conditions is that it's all about opportunity. We certainly weren't looking to move, but West Campus was a great opportunity to upgrade our space. Of course, a move like that doesn't happen every day, and many opportunities have come in the form of grants. Which brings us to our next stop. This room was renovated with funds from the Save America's Treasures program. This is vertebrate paleontology's newest collection room, and it happens to be Marilyn Fox's favorite room. Marilyn is our chief preparator of vertebrate fossils. Marilyn, what does a preparator actually do?
Well, Tim, we do a lot of things. So we take care of the collections. We go out and collect new fossils. And when you collect a fossil, it's encased in rock. So our main job is to uncover the bone and the information contained in the bone so that it can be used for research.
So what are you working on now, Marilyn?
So for example, right now I'm working mostly on a teeny tiny fragment of a lower jaw that we collected in the petrified forest. It's a new kind of animal that we don't know about. This is an interesting piece because it's got serrated teeth. That means it's a meat eater, but its jaw is only this big. We always have a variety of things that we're working on. And my colleague Brian, who's in the lab today, is working on a a block that was collected in the 1940s of an animal called Coelophysis, which is one of the earliest Triassic dinosaurs. So most of the animals in the Triassic were actually crocodile relatives. But what's really cool is in this giant block, there's more than one kind of animal. So there's the Coelophysis and unknown crocodile relatives. Tim, let me show you why this is my favorite room. So before we moved the specimens into this room, they were stored in what I think of as the archetypal dino star room, which was dark, dank, dusty, 1926 state-of-the-art shelving. And we've moved them into 21st century state-of-the-art shelving. So these shelves can open up with the touch of a finger. This is over 500 pounds on this shelf. And researchers can come in, it's well lit, it's light and bright, and they can measure, they can study these specimens without the danger of moving them around.
So what are we looking at here, Marilyn?
Well, this is the top of the femur, which is this thigh bone of Apatosaurus. And if you go up in the Great Hall, there's this giant bone lying on the Jurassic Island, and this is the other side of the leg. So we've had about 4 or 5 different researchers just in the last year here working on the relationships of these animals. So even though these were collected 150 years ago, they're used all the time by current researchers. Now I'll show you something else. So Tim, this is an example of 19th century shipping practices. This is something that was collected by J.B. Hatcher in the 1890s. And what they did in the field was they put it in this box with prairie grasses as a support. Then they put that in a horse and buggy, drove it across the Badlands, put it on a train, and that's how it got back to New Haven. But the grasses during all that time dried out, so they were offering no support to this poor bone, which pretty much got destroyed. So now what's actually more scientifically interesting about this specimen is the grasses.
Yeah, that's my understanding, Marilyn, that the grass can actually be used as a proxy now to predict what the environment was like during the 1890s.
It just means you can never know what's going to be useful for scientific research in the future.
If the story of the prairie grass tells us anything, it's that we can't always know what future generations will find important. So the Peabody isn't just about preserving the past for today. We're also about preserving the present for posterity. The Peabody's cryo collection is our newest collection. In the vats behind me are tissue samples of birds, fish, and mammals that were collected in the field by our researchers. These vats preserve the samples at temperatures below minus 310 degrees Fahrenheit. And because we're using liquid nitrogen, they can maintain those temperatures for up to a month if we lose power. The samples in this collection are available to scientists around the world, and their immediate use is to add genetic information to everything else we might know about an organism's evolutionary history. But because we intend to preserve these samples indefinitely, we can really only guess at what uses future researchers may find for these collections. But collections aren't just about having things, it's what you do with them. We're now in the class of 1954 Environmental Science Center, 2/3 of which is occupied by Peabody Collections and Labs. We deliberately designed this building to bring the collections and the people who study them into close proximity. The result is cutting-edge scientific research that attracts people from across campus and from around the world. Though in many cases, you don't actually have to come here to reap the benefits. So we're in the entomology collection with Nicole, and we were hoping you could tell us a little bit about the collection. What are you working on now, Nicole?
Well, we have a grant called the LEPNet grant, and this is a multi-institution opportunity for us to digitize all of our moth and butterfly holdings. That means we're going to be digitizing about 150,000 specimens over the course of the grant, possibly more. We have students coming in every day, maybe up to 10 students working at a time, and they read the data that's on the labels associated with our moth and butterfly specimens, and they enter that into our database.
So what's the goal of this, Nicole?
Well, we're trying to make everything that we have here at the Peabody accessible to anyone, anywhere. So it used to be that researchers or visitors may have to cross the entire world to get specimens that they're using for their research. Putting the data online means that any researcher who's interested in a certain group of insects can look at the data online and see whether or not the specimens they're interested in are here at the Peabody. Then they can contact us to get more information about those specimens. This research can be anything from research on conservation of environments to research on pollinators like bees. Everybody's interested these days in finding out why the bees are dying. So one of the areas of our collection that we digitized first was our bees.
So what are students and researchers using the collection for?
Well, so it may be that once they find that there are specimens here that they're interested in for their research, they need DNA sampling, or they need some kind of tissue sample. Sometimes we refer to this as grinding up their legs, because usually with insects, they'll remove a leg. And here we have a specimen. This is a swallowtail butterfly. And as you can maybe see, Tim, it's missing some of its legs. So instead of the ordinary 6, we have only 3 legs. Because insects have 6 legs, this gives us 6 opportunities for people to take tissue samples from a specimen. So that's often very useful for researchers. Although it damages the specimen, it's relatively minimal overall.
So why should we care about insects?
Insects compose some 80% of species diversity on Earth, maybe as high as 90%. They're most of what we have on Earth, and they impact our lives in every way. In addition to interesting and useful food sources, they provide research possibilities for medicine. They pollinate our food. So without them, we wouldn't survive for very long.
When museums started digitizing their collections, there was a concern that if people could view collections on their computers, museums would die off. In fact, it's been the opposite. More and more people are using the museum because of its online presence. So we're in invertebrate zoology with Dan Drew. Dan, what have you been working on lately?
So we're in the 3rd year of a National Science Foundation sponsored project to catalog and digitize our 50,000 plus microslide collection that has been acquired over the last 150 years by various Yale researchers, starting with our first curator and then all the way down to current research doing work on small crustaceans. So we're still making slides. The project was to catalog initially, gather all the metadata for the slides and enter it into our database. And part of the project is we bought a slide scanner to scan 2,000 of those slides at high resolution, and also low-res scans of as many of the slides as possible. We're probably going to approach somewhere in the neighborhood of 40,000 scans altogether. All of those scans will be freely available through the Yale Peabody website, and then the high-resolution scans are going to be available through our virtual microscopy website, which is geared towards education, particularly 4 through 8, high school, and even undergraduates and graduate students in colleges will be able to use the material.
So how is this better than traditional microscopy, Dan?
It's better than a traditional microscope in that the scanner allows you to look at the whole specimen at one time. A student looking under a microscope in the classroom would only see a small portion of that specimen. The scanner stitches it together and allows us to display the entire image. Also, microscopes are prohibitively expensive for many school systems, so we can produce the images that they would be seeing and provide them via the website.
So are there other advantages that have come out of this project, Dan?
For one, the best advantage for scientific researchers is the material that has been essentially lost to the scientific community for as long as 150 years is now available. Researchers can find the material, number one, know where to go and look for it, and be able to use the images instead of possibly having to come here, or we could take new images at higher resolution for them and send it to them.
So this is the Division of Invertebrate Paleontology, and we're here with Susan Butts, the collection manager for invertebrate fossils. So what have you been working on recently, Susan?
Well, Tim, we've got a grant from the National Science Foundation to look at the Western Interior Seaway, and the name of the grant is the Cretaceous World. So the Western Interior Seaway is a seaway that covered the interior of the United States and ran from Canada down to Mexico. And what we're doing is digitizing all the material. So the animals that lived in the Western Interior Seaway range from microfossils like foraminifera, zooplankton, to ammonoids, to very big clams in the Western Interior Seaway. And then we have the vertebrates like Hesperornis, which is in the Great Hall, Archelon, the giant sea turtle, which is in the Great Hall, and Zephactinus, the giant fish in the Great Hall. Now, we're working on this with collaborators from several different institutions. And by digitizing from all these different museums, we can get a better picture of what the ecosystem is. And then we can look at how that ecosystem responds over time to sea level changes. So we know that this time period, which is the time of T-Rex, is a time when there's big sea level changes. And that drives whole new changes in terms of biodiversity. So assembling this huge data set from all these different institutions gives a really solid base for studying what happens in response to climate change, in response to sea level changes related to climate, and then just how fossil communities interacted.
So what are the challenges, Susan, when you're dealing with this kind of material?
Well, this is a type of object that is a digitization challenge. So this is a cluster of fossils that are cemented together with minerals. And in the past, we would have cataloged this as one object, but now we have the ability to catalog each and every one of the clams in this fossil. And we have to develop pretty sophisticated workflows to do that. And what it involves is taking very high resolution images. And then we have a series of scripts that process those images, rename them as the specimen number for the Peabody Museum, and then introduce them to our database and attach them to this data for these specimens.
So who's doing this work, Susan?
So we do have a museum assistant who assembles material, but the people who do the bulk of digitization are Yale students. So we have 2 or 3 students each term that are digitizing all these materials. And over the course of the grant so far, which has been a year and a half, we've done about 70,000 objects.
From the Western Interior Seaway to microcrustaceans, swallowtail butterflies to DNA samples, Apatosaurus femurs to Panamanian pottery, and much, much more, the Peabody collections are at the heart of the museum's mission to tell the story of our planet and its people. We've been very fortunate over the year through grants, building renovation, and collection moves to not only improve the conditions of our collections, but to build on the museum's long legacy of giving the public, the Yale community, and researchers around the world as much access to our collections as we possibly can.